Hi, welcome to my YouTube channel BioExplorer. I am the Mujjaratna. This is an educational channel and this channel will be very useful to improve your biological knowledge. Today in this video we are going to talk about another type of cell division. It is meiosis. Meiosis is a type of nuclear division which gives rise to four haploid genetically non-identical daughter nuclei from a diploid mother nucleus. Meiosis is used for just one purpose in the human body, that is, the production of gametes, sex cells of sperm and eggs. The mother cells contain two sets of chromosomes, so they are diploid, and the daughter cells consist of only a single set of chromosomes, so they are called haploid cells. When a sperm and an egg join in fertilization, the two haploid sets of chromosomes form a complete diploid set. Meiosis can be seen in sexually reproducing organisms including plants, animals and fungi. Meiosis involves in two consecutive nuclear divisions as Meiosis One and Meiosis Two. Homolog pairs separate during Meiosis One and sister chromatids separate during Meiosis Two. In each round of division, cells go through four stages as prophase, metaphase, anaphase and telophase. Meiosis One. Before entering Meiosis One, a cell must first go through interphase. The cell grows during G1 phase, copies all of its chromosomes during S phase and prepares for division during G2 phase. Prophase 1. The cell enters to the prophase from interphase. Prophase is the longest phase in meiosis. The copied chromosomes condense into X-shaped structures so they can be easily seen under a microscope. Each chromosome is composed of two sister chromatids containing identical genetic information. Chromosomes also pair up. Each chromosome carefully aligns with its homolog partner so that the two match up at corresponding positions along their full length. A zipper-like structure called the synaptonemal complex, formed by specific proteins, holds the two homologous chromosomes tightly together. The pairing and physical connection of homologous chromosomes is called synapsis. During synapsis, part of the DNA molecule of non-sister chromatids of paired homologous chromosomes break, exchange and rejoin at corresponding points. This process, where the pair of chromosomes exchange bits of DNA, is called recombination or crossing over. You can see these points of crossovers under a microscope as chiasmata, where homologous chromosomes are linked together. Chiasmata keep the homologous chromosomes connected to each other after the synaptonemal complex breaks down. Commonly, each homologous pairs of chromosomes can have crossovers up to 25. The spots where crossovers happen are more or less random, leading to the formation of new remixed chromosomes. At the end of prophase 1, the nuclear membranes dissolve away, releasing the chromosomes. After crossing over, the spindle begins to capture chromosomes and moves them towards the center of the cell. Metaphase 1. The chromosome pairs line up next to each other along the center of the cell, which is called the metaphase plate. Each chromosome attaches to microtubules from just one pole of the spindle, and two homologous chromosomes are bound to microtubules from opposite poles. The centrioles are now at opposite poles of the cell. When homologous pairs line up at the metaphase plate, the orientation of each pair is random. That means they arrange randomly at metaphase plate. This allows for the formation of gametes with different sets of chromosomes. Anaphase 1. Kinetochore microtubules of the spindle get shortened. Homologous pairs of chromosomes separate, and each chromosome of the pair is pulled towards the opposite pole. Sister chromatids of each chromosome remain attached at the centromere and move as a single unit towards the same pole. This is different to what happens in mitosis and meiosis 2. Telophase 1. Here, chromosomes have arrived at opposite poles of the cell. At each pole of the cell, a full set of chromosomes gather together. Nuclear membrane forms around each set of chromosomes to create two new nuclei. Nucleoli reappear. A spindle is broken down into its monomers. Chromosomes decondense into chromatin. A genetically non-identical haploid two daughter nuclei are formed within one cell. Cytokinesis usually occurs simultaneously with telophase 1. Genetically non-identical haploid two daughter cells are formed. In animal cells, cleavage furrow is formed, and in plant cells, a cell plate is formed. But in some cells, cytokinesis is skipped since cells will soon go through another round of division, that is meiosis 2. Meiosis 2. This is the second meiotic division. It usually involves in separation of sister chromatids. Mechanically, this process is similar to mitosis, though its genetic results are fundamentally different. The end result is production of 4 haploid cells from the 2 haploid cells produced in meiosis 1. Meiosis 2 is a shorter and simpler process than meiosis 1. Cells move from meiosis 1 to meiosis 2 without copying their DNA. Those cells that enter meiosis 2 are haploid and have just one chromosome from each homologous pair, but their chromosomes still consist of 2 sister chromatids. Prophase 2. Chromatin fibers condense and produce X-shaped chromosomes with 2 sister chromatids. They can be easily seen under a microscope. Nuclear envelope breaks down into fragments, releasing the chromosomes. Nucleolus disappears. The centrioles duplicate and start producing spindle apparatus. During the late prophase 2, centromere of the chromosomes are moved to the metaphase 2 plate. Metaphase 2. The centrioles are now at opposite poles in each of the daughter cells. The chromosomes line up individually along the metaphase plate. The 2 sister chromatids of each chromosome are captured by microtubules from opposite poles. Due to the crossing over in meiosis 1, the 2 sister chromatids of each chromosome are not genetically identical. Meiosis 2 usually takes place in the perpendicular direction of meiosis 1. Therefore, metaphase plate of meiosis 2 is perpendicular to the metaphase plate of meiosis 1. Anaphase 2. Due to the breakdown of proteins attaching the sister chromatids, they are separated at centromere. As a result of shortening of kinetochore microtubules, the sister chromatids are pulled towards the opposite poles of the cell. The separated chromatids are now individual chromosomes. Telophase 2. At each pole of the cell, a full set of chromosomes gather together. Nuclear envelope and nucleolus reform. Chromosomes decondense into chromatin. The spindle disassembles. Genetically non-identical haploid 4-daughter nuclei are formed from one parent cell. Cytokinesis. Cytokinesis splits the chromosome sets into two cells, forming non-identical haploid 4-daughter cells. These daughter cells are not even identical to their parent cell. Significance of meiosis. Maintains the constant number of chromosomes through generations in sexually reproducing species. There are 46 chromosomes in the nucleus of a human diploid cell. Through meiosis, gametes, example sperm and egg, with 23 chromosomes are produced. Those gametes fuse by the process of fertilization, forming a zygote, which has 46 chromosomes. So the number of chromosomes of a species remains constant from generation to generation. Produce new genetic variation leading to evolution. Genetic variation occurs due to crossing over, recombination and independent assortment. The points where crossing over and exchanging genetic material are chosen more or less at random. This helps in producing gametes with different genes. The random orientation of homologous pairs of chromosomes in Metaphase One allows for the production of gametes with different variations of genetic materials. So that's all today. Hope you enjoy this video and gain lot of interesting facts about meiosis for your biological knowledge. So please subscribe this channel and click the bell icon. See you in the next video.